Okay, so we'll be looking at bacteriophages, right? You have been introduced to bacteriophages before when we look at bacterial transduction. Right? So in this topic, where we are focusing on the viruses, we'll be looking at the bacteriophages in more details. Okay, so firstly, the structure, right? It looks a bit like an alien. Okay, so there is this head structure, which is basically the capsid coat, which contains the genome of the bacteriophages. The genome is in the form of linear double-stranded DNA, usually one molecule, but some phages may have more than one molecule. Okay, and below the head, there's this tail structure, which is basically a hollow tube made up of proteins. The hollow tube of the tail, right, is on the outside there is this contractile sheath. Okay, and at the end of the tail, there's this base plate and usually some tail fibers as well. Okay, so the base plate and tail fibers function to bind to specific receptors on the bacterial host cell that the virus is going to infect. The contractile sheath, right, would contract, as the name suggests, and that would drive the tail into the bacteria. It will puncture the bacterial cell wall and drive the tail into the host cell. All right, and because it's a hollow tube, right, so the linear double-stranded DNA is able to pass through this hollow tube of the tail into the bacterial host cell. Okay, we'll look at it in more details a bit later when we look at the reproductive cycle. Okay, so in your syllabus, you are introduced to two types of bacteriophages, right? The T4 bacteriophage, which reproduce by lytic cycle, and it is known as a virulent phage, and also the lambda bacteriophages, which reproduce both lytic and lysogenic cycle. It is known as a temperate phage, all right? And in terms of structure, they are pretty similar. The T4 is slightly larger, right? So even though on this diagram it looks like they are the same size, right? It's not, right? The T4 is larger than lambda. And also in terms of the structure as well, tail fibers, right? A lot of tail fibers for the T4 phage, but like usually one short tail fiber for the lambda phages, right? But such small details you don't need to be too concerned with lah. Okay, so anyway, let's look at the reproductive cycle for the T4 phage, which reproduces by lytic cycle only. Right, so the first step, right, is attachment that involves the tail fibers and the base plate. They will bind to specific receptors on the host cell. After that, the contractile sheath of the tail, right, would contract, and that would drive the tail into the bacterial cell past the bacterial cell wall. Right, and because it's a hollow tube of the tail, so the viral DNA, the linear double-stranded DNA, is able to pass into the bacterial host cell through this hollow tube, and that would be the start of viral infection, and that is start of what we call the eclipse period. We'll look at it in more details a bit later in terms of all these different periods. Alright, so anyway, the viral DNA has now entered the host cell, and this is a diagram showing you the entry of the DNA, right? You can see that the DNA is entering into the bacterial host cell, and the capsid head, right? The head structure of the bacteriophage will become empty once the DNA has all entered into the host cell, right? So this empty capsid head and the tail, right? This empty phage will be left outside the bacterial host cell. Okay, so the only thing that enters is the viral DNA. Okay, so once the viral DNA is entered the host cell would replicate this viral DNA, and also the ribosomes, the RNA polymerases, right, would carry out protein synthesis using the viral DNA as template for transcription, and then later the viral mRNA as templates for translation. So one of the viral proteins that are made, right, is actually an enzyme that would degrade the bacterial DNA, right? So that would chop up and hydrolyze the bacterial chromosome and also plasmids as well. right? So that would prevent the bacterial host cell from undergoing its own synthesis, be it DNA replication or protein synthesis. So all the resources, raw materials, the ribosomes, the enzymes, right, the energy, everything is devoted to the replication of the viral DNA as well as the synthesis of the viral proteins. So this is what we call, right, the viruses taking over, right, the host machinery. Right, so the host machinery is no longer making its own DNA and proteins, it is making the viral DNA and viral proteins. Right, so once the different components of the viruses, the DNA, the proteins, has been made, right, they would be assembled into new phages inside the bacterial host cell. Right, so the capsomeres would form the capsid coat, which would be formed around the viral DNA, and then the other proteins that forms the tail fibers, right, the base plate, the contractile sheath would all assemble together, right, into new phages. So that's the assembly stage. Okay, so you can see on this diagram, right, the a lot of viruses, a lot of the bacteriophages, right, found inside this bacterial cell. You have some outside as well, so those might be viruses that are trying to infect this bacterium, or they might be the empty capsid, right, that has been left behind after the entry step. All right. So the last step is the release of the new viruses, and that involves another viral enzyme that is made during the synthesis stage, right? So this is a viral enzyme that is called a lysozyme, and it breaks down the peptidoglycan cell wall of the bacterial host cell. So without the cell wall, right, to protect the host cell, the host cell will lyse. Right, so usually the bacterial host cell will be found in an aqueous environment, right? So water usually would enter the bacterial cell via osmosis, right? So the cell wall is there to prevent the bursting of the cell. So now that the cell wall is hydrolyzed by this lysozyme, right? So then the bursting due to the osmosis of water into the cell would occur, and that's what we call the lysis of the host cell. Right, so once the host cell lies, right, it breaks apart, right, then the new viruses will be able to escape, and that's the exit or release step. So this is known as the burst period, where we see a very fast increase in the number of bacteriophages because of this sudden release, right, when a host cell bursts or lies. All right. So when we plot the numbers of phages, the bacteriophages, right, versus time, right, this is where we get all the different periods, right? So this is the start of the replication cycle, where the virus, the bacteriophage, attach and also insert, or rather transfer the viral DNA into the bacterial host cell, right? This is when we see the appearance of the first phage. That's why it's for the curve for total phage, it starts to increase, right? And but then if you look at this graph, this curve here, right? If you can follow my cursor, or maybe make it clearer, yeah? Okay. So this curve, this dotted line, shows extracellular phage, right? So this is total phage, this is extracellular phage, and at this point, there's no extracellular phage yet, which means this total phage, right, the appearance of all these phages, right, is still inside the host cell, right? So between the start of infection to the appearance, the assembly, right, of the new viruses inside the host cell, this is the eclipse period, right? So now we have this curve that shows an increase in the extracellular phages, right, which means that the bacteriophages, extracellular, right, means they have been released from the host cell. And so this is the start of the exit or release stage, right? So from the start of infection all the way to the start of the exit stage, right? This is known as the latent period for bacteriophages, right? So this is like the end of release, where the number of phages no longer increase, right? So all has been released. Okay, so the amount of release is known known as the burst size, right? And the period where we have the first phage released all the way to the last phage being released, this is known as the rise period. Okay, so some periods when we break up the different stages of the reproductive cycle of the bacteriophage. So putting it into the description of the reproductive cycle, right? Again, eclipse period, there's infection as well as the replication stage, but not yet assembly, right? Then we have the end of the eclipse period and the rest of the latent period. That's when assembly has occurred but not yet exit. And the rise period will be the exit. So you can match the graph and match the different periods, right, to the reproductive cycle of the bacteriophage. All right, so you have learned lytic cycle now, and you have learned generalized transduction before. And if you recall, generalized transduction is due to the lytic cycle, right? So generalized transduction is the transfer of bacterial DNA, right? So how does the lytic cycle give rise to this generalized transduction, right? Okay, so remember that the bacterial DNA is broken down by viral enzymes, and some of these bacterial DNA fragments might be assembled into the bacteriophages by mistake. And so it should be the viral DNA that's assembled, right, inside the capsid head, right? But then by mistake, there might be fragments of bacterial DNA being assembled instead, right? So these bacterial phages that's newly assembled, right, they may actually carry the bacterial DNA instead, right? But these new bacteriophages, they still have the base plate, right? They still have the tail fibers, right? So once they are released, they can still infect another bacterium, but instead of transferring viral DNA, they will transfer the bacterial DNA. All right, so that's the generalized transduction. Okay, so I hope you understand generalized transduction better now, having looked at in more details lytic cycle, because I understand that quite a number of you actually did not really understand or cannot really visualize the generalized transduction before, and I have a lot of questions from my own students as well. So I hope with this more detailed description of lytic cycle, you understand better generalized transduction. Okay? So next we'll be looking at lambda phages, which reproduce by both lytic and lysogenic cycle. Okay? So the lysogenic cycle is the process where the genome, the DNA of the virus, is replicated, but the whole cell does not die. Okay, so we'll look at why later. All right. So the first step is still entry, right, which is similar to T4. The difference is, of course, your lambda phage only has one short tail fibers, right? So much of the binding to the specific receptors is responsible or is due to the base plate lah, because the one short tail fibers doesn't really help much in terms of the binding. Okay, but other than these small details, the entry the attachment is kind of similar. So there will still be the contraction of the contractile sheath, right? The hollow tube of the tail will still puncture into the bacterial host cell. The viral DNA will still enter the bacterial host cell. All right, so then this is the difference, right, in terms of the lambda phage versus the T4, right? For the lambda phage, right, the viral DNA actually circularizes first. So it's not seen in T4. Okay. So from after this step, right, the bacterial reproductive cycle can go two ways. It can either just continue with the lytic cycle, so that's where we have the usual replication stage. The bacterial DNA is going to be broken down by enzyme. There's assembly of new viruses and released. All right, so that's the usual lytic cycle. However, right, for the lambda phage, it can also enter the lysogenic cycle. This is when the viral DNA actually gets integrated into the bacterial chromosome. All right, So once integrated into the bacterial chromosome, the viral DNA is now called a prophage. And the infected bacterium that carries this prophage is known as a lysogen. That's why the name lysogenic cycle. Okay, so this viral DNA or prophage now, right, is inserted into the bacterial chromosome. There's not going to be protein synthesis except for one viral repressor protein. Right, so we call what's a repressor protein. Right, so this viral repressor protein will be made. Right, so there will be transcription of the gene coding for this repressor protein and the translation of this protein, and this protein would in turn represses the expression of the other genes of the virus. And so there's not going to be the enzyme to break down the bacterial DNA. There's not going to be making of capsomeres and so on. Right. So as a result, right, your viral DNA just stays silently, right, in the bacterial chromosome. Right. So when the bacterial host cell divides, it will replicate its bacterial DNA, right, which includes the viral DNA, right? So the viral DNA gets replicated as well. It stays as part of the bacterial chromosome, right? So now instead of one infected cell carrying the prophage, now you have two infected cells carrying the prophage. So this stage, this is the lysogenic cycle, right, where we have again replication of the viral DNA, but the whole cell does not die, right? The whole cell is able to continue to divide. So this is the lysogenic cycle. All right. So of course, the bacteriophage will not stay in this lysogenic cycle forever, right? There will be induction back to the lytic cycle. It can be due to some form of external stimuli, or it may be a response to unfavorable conditions that may kill the bacterial host cell, right? Or it may be just spontaneous, right, for no reason, right? So anyway, once lytic cycle is induced, then it will just go back to lytic cycle. The first step is for the viral DNA or prophage to be excised from the bacterial chromosome. Right, so now it comes out from the bacterial chromosome, as you can see. So once it comes out, then the rest is the same as the lytic cycle, right? Then you have the replication stage, right? Protein synthesis, assembly, and exit. All right. So this is how the lambda phage or similar temperate phages, right, reproduced by both the lysogenic and lytic cycle. Okay. Right, so again, back to the bacterial transduction, right? So you learned that the lysogenic cycle is involved in specialized transduction. Right, so now that you have looked at what is the lysogenic cycle in more details, can you then link and therefore understand specialized transduction better? Okay, so you learned that, right, when the prophage is induced to exit the bacterial chromosome, right, that's when the lysogenic cycle stops and the lytic cycle is induced, right? By mistake, instead of only the prophage being excised or only the prophage coming out from the bacterial chromosome, there might be some bacterial DNA next to the prophage that will be coming out together with the prophage. Right, so this is a mistake again. Right, so in this case, your new bacteriophages would all contain the prophage as well as these pieces of bacterial DNA next to the prophage. Right, because when this piece of DNA comes out, right, the prophage and the pieces of bacterial DNA, this piece of DNA gets replicated, right? And that becomes the genome for all the new viruses, right? So now all the new viruses that's made would contain both the prophage and also this adjacent bacterial DNA. Right, so obviously when these bacteriophages are released and they infect another bacterium, then they would transfer the viral DNA together with the bacterial DNA into the next infected cell. So this is specialized transduction. Specialized because the location where the bacterial DNA is inserted into the bacterial chromosome is not random, right? It's always kind of the same site, right? So whenever the adjacent bacterial DNA is pulled out, when the prophage leaves the bacterial chromosome, it's kind of the same, or roughly the same parts of the bacterial DNA that's removed together with the prophage. Right, so that's why it's specialized transduction. You're always transferring kind of the same pieces of bacterial DNA. All right. Okay, so I hope you understand bacteriophages and also revise your bacterial transduction. All right.